We need to talk about the biological importance of lipids, and they can list them and discuss, at least give a short explanation of each important you mentioned or what significance. For example, here we said lipids are very important in terms of being the dietary component. So you find the lipid input, and they play a very important role either because they have a caloric value. For example, in triacylglycerols, triacylglycerol we say comprises of fatty acids, and these fatty acids are very important when it comes to energy productions. Also, some of these lipids play a very important role as biological because they have biological activity like omega-3, omega-6 fatty acids. Those of us that will also talk about fatty acids will definitely tell us what are omega-3, what are omega-6, how important they are to the extent that the deficiency of omega-3 and omega-6 can manifest to some conditions which they need to cover that. And individually, we also need to know that. And we also need to know that this lipid serves as energy storage molecule, just as we mentioned in the triacylglycerol usually found in adipose tissue, that unlike a protein, when you take a food containing a protein, it will be broken down to amino acids, and this amino acid will be utilized by the body. And excess of the amino acid cannot be stored by the body. Therefore, the excess amino acid can either be converted to glucose. The other time we talked about the fate of amino acid as either glucogenic. So those amino acids that were excess in the body after taking food are either converted to glucose, we call them glucogenic amino acid, or converted to ketone bodies or fatty acid called ketogenic amino acid. Why? Because they cannot be stored in the body. Meanwhile, when you have a carbohydrate, when you take excess of carbohydrate, that carbohydrate can actually be stored in your body. The same thing with fat also, excess of it can be stored in the body. And we said the excess fat is stored in form of mostly triacylglycerols in adipose tissues. And we also made mention in some part of this class that the lipids form the major component of cell membrane, where we mention basically phospholipid and some other type of glycolipids. We also said they facilitate the transportation of substance, like transportation of water-insoluble molecules into the cells. And we made mention that some molecules doesn't have to pass through some membrane transporters. They can easily dissolve in the lipid bilayer membrane and then transverse themselves into the, or fuse themselves into the cells. The lipid can also serve as precursors of some part-soluble vitamins. For those of us that talked about steroid hormones, they will see some steroid hormones like progesterone, testosterone, androgen, which are basically a lipid in nature, in a chemical nature. And we also know that they're involved in intra and intra signaling process. Some of us who talked about some of these prostanoids like prostaglandins and leukotrienes, and how they play a very important role as intracellular messengers. We also know that part of the subcutaneous tissues, which serve as a tissue insulators, we know that some individuals that accumulate part, usually this part is actually stored in the subcutaneous tissues. And these subcutaneous tissues, what do? They basically form a kind of an insulator, trying to protect the organs from any damage. One we can easily notice is how fat can be deposited just beneath the skin. So for some individuals that are very chubby, what is basically behind their skin is the accumulation of these lipids, and why are these lipids basically deposited there is to serve as insulators, to serve as protectors to these organs. So this is basically what I'm expecting from, although I have elaborated, so this is just for that to tell you that it's very important. But for other ones, I don't need to. Okay, now classification of lipid. For those that are supposed to talk about lipids, they would definitely give the classification where we mention simply here as lipid have been classified based on the number and the type of component present. We have a simple lipid, which can be fat, oil, and waxes. We have a complex lipid, and the complex lipid, how do they differ with the simple lipid? Basically, due to the presence of the phosphate, sugars, pinkosine backbone, which definitely they will explain that. And of course, we talked about some of them like phospholipids. They contain a phosphate in addition to the simple lipid. We also say sugar. They contain a sugar in addition to the simple lipids where we mention glycolipids. And we also have drive lipid. This drive lipid, basically the hydrolysis of a complex lipid. So you have some component. What are the component of a simple lipid? We said they have fatty acids. So you can have the fatty acid and the glycerol backbone, etc. So we also have a group that will talk about fatty acids and their classification. So basically here what we're expecting from you is you tell us what are fatty acids. You tell us the classification based on saturation level. We have saturated fatty acids. We have unsaturated fatty acid. You can go ahead and tell us more about unsaturated, for example, the monounsaturated fatty acid and the polyunsaturated fatty acids. One of the important things you need to tell us about the polyunsaturated fatty acids are the omega-3 and 6 fatty acids. Why are these omega-3 and omega-6 fatty acids were considered essential? And why deficiency in this omega-3 and 6 is or lead to some symptoms. So you have to tell us that. And we have some that we talked about eicosanoids and their classes, which basically are two. We have a life. Someone is talking.
Sorry, sir.
Okay. So we have a eicosanoid. So you talked about the two classes of eicosanoid. We mentioned the local hormones. You have prostanoids like prostaglandin, thromboxanes, and prostacyclins, and how this play a very important role as hormones, usually in pain and some other signaling process. And you also talked about at least give one example of the prostanoids, and at least also give one of the linear eicosanoids like leukotrienes and lipoxins. All these things I'm saying with regard to the assignment, those people that are supposed to cover that. And another group also talked about triacylglycerols and their composition. So you just tell us what is triacylglycerols, and you tell us that triacylglycerol comprises of glycerols and then the fatty acids attached to the carbon backbone of the glycerols. And you tell us the composition of the fatty acid and how does the composition of the fatty acid affect the physicochemical property of triacyl glycerols. And you tell us, okay, that is that for that. And then we have another group that we talked about steroids. So you tell us the classification, the bile acids and the steroid hormones. And another group we talked about lipoproteins. So they will give us the classification of lipoproteins, which include the high density lipoproteins, low density lipoproteins, very low density, and chylomicrons. And to what significance are these lipoproteins, most especially in clinical terms, and the clinical relevance, how these lipoproteins they play a very important role in the pathogenesis of many disease, especially the heart-related disease. Are we together? Class, are we together?
Yes, sir.
So basically, for those of us that have their
Yes, sir.
for those of us that have their own group assignment within this study session, this is basically what I'm expecting. So if you have done anything that is out of this, please, you need to go back and look at it and at least pay much attention to those things I mentioned. And this is general to everyone that please, you have to pay attention to classes of I made mention some of these things that you have to pay attention to. What are the classification of lipids? Do you know them? The simple lipid, the complex lipid, the dry lipid, because these are the basics and the most important things. What are fatty acids? What is the difference between the saturated fatty acid and the unsaturated fatty acid? Why do we have essential fatty acids and which ones are not essentials? Why are they called essentials? What are the symptoms related to those essential fatty acids? Some of these things are the things that you have to pay much attention to. Okay. So I think we've made a point with regard to the assignment of some of us. So I think when we reach because some other group will have their assignment under study session 4 and study session 5. So when we reach study session 4 and study session 5, I will also highlight, give more information on what I'm expecting from them. Is that clear?
Yes, sir. Yes, sir.
So
Yes, sir.
Under study session 3, study session 2, we are going to talk about the structures of DNA, structure of DNA and RNA. Under this, the main content of this, we will talk about the structure of DNA. We talked about description of the duplication and RNA synthesis, and we talked about importance of some synthetic nucleotides. So basically, summary of our previous class, I think, which study session is that? So I cannot see the heading of this. Can somebody read the heading of this slide? Can somebody read the heading of this slide? Can you hear me?
So did you make the headings like in tabular form or the?
Oh, okay. Now, I can see it. No, something just covered my listen. So this is just a summary of module 1 study session 3. I think in module 1 study session 3 is where we talked about the chemistry of nucleic acid, where we mentioned of all these bases and how this basis comes to give what we call the nucleosides and the nucleotide, right? Did you remember?
Yes, sir.
Yes, and in that we were able to while explaining the bases found in the DNA, the ones found in the RNA, the sugar and the phosphate, we were able to give some differences between the RNA and the DNA. But basically at the end, we made mention that in the DNA, what we have is the deoxynucleotide, while in the RNA, what we have is the nucleotide, or let me say deoxyribonucleotide, while in the RNA, we have ribonucleotide, basically, okay? So what is important here is that, are you with me?
Yes, sir.
So in the previous class,
Yes, sir.
we learned that 2 deoxyribonucleotide can be bonded to each other through a bond we call phosphodiester bond, right? And we have what we call a dinucleotide. And how does this dinucleotide is formed? It is formed in such that a phosphate group in one of the nucleotide is attached to carbon 3, are we together? Of the ribose sugar. And carbon 3 of the ribose sugar. Remember how the naming of carbons in ribose sugar is what? We say they are primed. We have carbon 1 prime, carbon 2 prime, carbon 3 prime, up to carbon 5 prime. Is that clear?
Yes, sir.
Yes.
Yes, sir.
So when we have a phosphate group, and remember while we are talking about the formation of nucleotide, we say a nucleoside contains the sugar and the base. By the time we attach the phosphate group, we have what we call a nucleotide. And we said the phosphate group have an option, most especially in the case of a DNA, deoxy sugar. Deoxy sugar does not have a hydroxyl group at carbon 2. So at carbon 3, there is hydroxyl group, and also carbon 5, there is hydroxyl group. So the phosphate have an option of either attaching to carbon 3 or attaching to carbon 5. But in DNA, the phosphate for the formation of nucleotide, the phosphate is attached to carbon 5. Meanwhile, we have a hydroxyl group at carbon 3. So meaning that another phosphate, there is a chance of another bond formation at carbon 3. So when we have the two nucleotides, we know they have a hydroxyl group at carbon 3. So a phosphate group of one nucleotide can form another bond with the carbon 3 of another nucleotide. That bond is called phosphodiester bond. Are we together?
Yes, sir. Yes, sir. Yes, sir.
Okay. So we have this. Now we know what is a phosphodiester bond, and we know how it is formed. All right. So now just assume you have more than 100 nucleotides. This nucleotide can be bonded to each other through the phosphodiester bond. And we call it a polynucleotide. Is that clear?
Yes, sir.
So when you have a polynucleotide, this polynucleotide have a two end. Just like while explaining the amino acid sequence, we talked about N-terminal and C-terminal. In the case of DNA, we have the 5' end and the 3' end. I think I don't need to explain why we have a 5', why one is called 5', the other one is called 3'. Do I need to explain that? Because when you look at the nucleotide, just look at the post reaction here where we have a two nucleotide forming a phosphodiester bond. And on one nucleotide, we have a phosphate group attached to carbon 5, and then we have an OH group at carbon 3. So also the same thing in all the nucleotides. So in the formation of phosphodiester bond, it involves, for example, when you look at this first, I think you can, if you can see my cursor, you can see my cursor here. We have the OH group at a 3' carbon here, And we have this phosphate attached to carbon 5. They are the ones that are involved in the phosphodiester bond. Meanwhile, you have at this 5' carbon, you have a phosphate, 3, and you have another hydroxyl group here at the other nucleotide. So this end is one end. We call it 5'. And here is the other end. We call it 3'. So if another nucleotide come to join and form a phosphodiester bond like this, until when we have several of them forming, at the end you have one end having 5' free, and the other end will have a 3' primer. Is that clear? Is that clear?
Yes, sir. Yes, sir.
Okay. Complementary base pairing. Now we made mention that in DNA, in a DNA exists in form of what you call a double strand. Unlike in the case of RNA, RNA is a single strand. DNA is a double strand. So we have seen how a polynucleotide, a single strand, is formed just as in here. We can see a single strand here by the right. This is a single strand of the, we have a nucleotide like adenylate, cytidylate, guanylate, and thymidylate. Forget about that U in a bracket, thymidylate. We have this polynucleotide. How are we having the two strand? Are we having the two polynucleotide coming together to form a double strand? In either way, whatever happens, we'll see how that is formed. But what we have in the case of DNA, we don't have only polynucleotide strand, only one polynucleotide strand. Rather, we have a two polynucleotide strand joining together to form a double strand. How does the double strand is formed? The double strand is formed through what we call complementary base pairing. This is simply to say one nucleotide pair with another nucleotide to form the complementary base pairing. And the complementary base pairing is done in such a way that a purine and pyrimidine will bind to each other to form a base pair. Not two purine will bind to each other. Not two pyrimidine bind to each other. For example, now let's say in DNA. In DNA, we know we have 4 nucleotides. We have adenine, we have guanine, and we know that adenine and guanine, they are purines, right? Right?
Yes, sir.
Yes. We have adenine, we have guanine, and these are the only purines in DNA. While the pyrimidines in DNA, we said we have a cytosine, we have a thymine. And we said this complementary base pairing only occur between the purine and a pyrimidine. So you can have one purine form a complementary base pairing with another, with a pyrimidine. But you cannot have a purine forming a complementary base pairing with another purine, say adenine pair with guanine. It's not possible. So you can have an adenine. So what happened basically is adenine pair with thiamine. So we have adenine as purine, thiamine as pyrimidine. So when you have an adenine pairing with thiamine, they form a hydrogen bond, 2 hydrogen bond. Then we can see from the structure A we can see 2 hydrogen bonds formed between adenine and thymine. While in the case of guanine and cytosine, we have 3 hydrogen bonds formed. Is that clear? So in summary, in complementary base pairing, the complementary base pairing is formed between the nitrogenous bases of a nucleotide. How does it form? It's formed between purines and pyrimidines, where we have adenine binding or forming a 2 hydrogen bond with thymine, while a guanine forming a 3 hydrogen bond with cytosine. Is that clear? So
Yes, sir.
when you have a single strand like this, the adenine here, you will have another, what will bind to it to form a complementary base pairing? Class, are we together?
Yes,
Yes,
sir.
sir.
When
Yes,
you
sir.
look at the polynucleotide here, the first nucleotide we have is adenine, right?
Yes, sir.
With the E indicating adenine. Forget about the other side is showing you, the one in yellow is showing you that this is the ribose sugar. And the most important thing here is we are trying to show you how the phosphodiester bond is formed. Therefore, we just indicate something like the yellow to indicate the ribose sugar, and this E to indicate adenine. This C indicate cytosine, the ribose sugar, and then the phosphodiester linkage. So now we have the first one as